Dear students, today we are going to study the fluid mosaic model of the plasma membranes. Various models have been put forward to explain the structure of the plasma membrane, and the most acceptable one is fluid mosaic model, which was put forth by SINGH and Nicholson in 1972. Now, what is the structure of this plasma membrane? It is largely constituted by lipids and proteins, which are arranged in a mosaic fashion. They are not arranged in a peculiar or particular kind of fashion, but they are arranged haphazardly. And since they have non-covalent interactions between them, the lipids and proteins could able to move inside the plasma membrane, and this is called as translational movement of lipids and proteins. And it is because of this non-covalent interactions present between proteins and lipids, the plasma membrane structure is a quasi-fluid kind of structure. And these non-covalent interactions are ionic and hydrogen bonds. Had they been held by covalent bonds, the structure of plasma membrane would have been a rigid one. But since ionic and hydrogen bonds, they are prevalent in the plasma membrane, the fluidity of plasma membrane can be explained by presence of these two kinds of bonds, which are not so rigid. The molecules which are present in the plasma membrane, they are amphipathic in nature. Amphipathic means having two kinds of characteristics, and that is they are having hydrophilic and hydrophobic parts both present in the same molecules. The intrinsic proteins are also amphipathic. That means the hydrophobic part of the intrinsic or integral proteins, they remain embedded in the lipid bilayer. However, the protruded part of these these proteins are hydrophilic in characteristics. So this arrangement explains the presence of various active sites in the on the plasma membrane. This model is supported by freeze fractured membranes, and this structure has been drawn from the RBC plasma membrane. And you can cite the red color proteins, which are integral proteins. They have two kinds of domains. One is hydrophilic domain, which remains outside the lipid bilayer and naturally inclined towards the extracellular fluid, which is watery or polar. However, the alpha helix or hydrophobic part of the of the integral protein or intrinsic proteins remain embedded in the lipid layer, and it has amino acids which are hydrophobic in nature, and this hydrophobic domain is very important for the integration of protein as such in the plasma membrane. And you all know that how very important the proteins are in the plasma membrane. They play a very vital role. Now, some cholesterol molecules are also present in the plasma membrane. This was the most acceptable model, which was posited by SINGH and Nicholson in 1972. And apart from these, certain other models have also been proposed, and one important one is unit membrane concept, and it was given by Robertson. What he stated that if we see the plasma membrane under the microscope, electron microscope, the cell membrane appears like two dense osmophilic zones separated by a clear zone, as you can see in the slide, which depicts the lipid bilayer between two protein layers. These yellow colored are two protein layers. One is towards ECF and another is towards the ICF. And in the protein layer, you can cite the lipid monolayer towards the ECF and another lipid monolayer towards the ICF. And in all, this membrane is 75 angstrom thick. What he wanted to say is that lipid is the bimolecular lipid layer is sandwiched between two protein layers, but this theory is no more accepted these days because you have studied that the lipids and proteins, they are in translational motion, and there is the lipids and proteins, they are not arranged in a typical fashion. In fact, they are haphazardly and mosaically arranged, as it was portrayed in the fluid mosaic model. Another model is the sandwich model, and this is called as protein lipid protein model. This was put forth by Denali and Davison in 1935. You can see in the slide that the model has two lipid bilayers, two lipid layers, having polar groups and non-polar groups. Non-polar groups naturally inclines away from the ECF and ICF, whereas this model says that the proteins, they are laid on the top of these top of the bilayer, which is discarded now, and these proteins are tangentially arranged and are in contact with the lipid bilayer. This was given in 1935, and in between there are some spaces which are called as pores, and these are called as polar pores because these contains polar proteins. So this was another model which was discarded later on, and the model which is most important to you is fluid mosaic model. And many a times unit membrane concept is also asked in the examination, but the most important one is fluid mosaic model. Now we have to study in detail the proteins which are present in the plasma membrane. These proteins which are present in the plasma membrane, they have been classed into two kinds: integral proteins and extrinsic proteins. The integral proteins, as I've told you in the beginning of this lecture, contains hydrophilic domain which contains polar amino acids, whereas the transmembrane helix is non-polar region embedded in the lipid bilayer, forming hydrophobic interactions with the fatty acyl chain. The and the hydrophilic domains contains polar amino acids, whereas the hydrophobic part which remains embedded in the lipid bilayer contains hydrophobic amino acids which are in interaction with the fatty acyl chains of the lipids. The hydrophobic domain has an importance in the sense that it is helpful in the insertion of this integral proteins in the lipid bilayer. So the examples of the integral proteins which are present in the plasma membranes are porins, cadherins, integrins, selectins, band 3 proteins, glycophorins, sialoglycophorins, and there are certain antigenic determinants also. So you can sense that how very important the plasma membrane and its protein are for the recognition of the same uh, part of the plasma membrane, or it is what I wanted to say is कि antigenic determination means they discard or accept the organs or blood transfusion, as you would study in detail in the higher classes. So students, this is all in this lecture. In the next lecture we'll study the rest part of the integral proteins and the extrinsic proteins as well as the lipids. Thank you very much.